Hello, in today's session, I want to discuss with you anticoagulants used especially in hematology laboratory. We want to see how can we preserve blood which is collected to keep in the same way it was in the body. So we want to look at different anticoagulants or the preservatives that we can use to make sure the blood remains in its normal way, the cells remain in their normal morphology without alteration. And these anticoagulants which we are talking about, what are they? So the anticoagulants, these are chemical substances. They are chemical substances, and these chemical substances, they have ability to stop blood from clotting. So they stop blood from clotting. So their major aim is to stop or to reduce the rate at which the blood clots. But the major aim is to stop blood from clotting. That is the anticoagulants. So they are chemical substances, they can be industrial, and they are made industrially, or there are some which are natural. And these are used to preserve blood. And what is the reason for applying or for preservation? The significance of preserving blood. What is the significance of blood preservation? That after phlebotomy, or that is the process of collecting blood, why do we need to preserve it by combining it with anticoagulants? And we are seeing one of the major significance is to prevent clotting of blood, or we can say is to stop blood from clotting or to prevent clotting. That is one of the major aim. And another major aim, we are preserving the morphology. We are preserving the morphology of cells in the blood. Morphology of cells to remain the way they are in the body. And another thing, we are making sure the substances which are depleted, like glucose, is preserved in the normal concentration. So that is preventing blood clotting, preventing morphology, and sometimes we are preserving chemical components in the blood like glucose from being depleted. So those are some of the significances why we need to clot. And still we can use preservatives for storage to increase the shelf life. To increase the shelf life of blood collected in the vagina, that is to enable storage. Because whenever we draw this blood from the body and it stays more than 6 hours without a preservative, we lose their morphology. So we cannot stay them for longer. But now these days, because of anticoagulants, we are able to store our blood collected in the laboratory for even years because of the anticoagulants. And after seeing the significance of preserving blood, now we need to know the anticoagulants. And I'm going to tabulate these anticoagulants. Allow me to remove this, whereby we are going to form a table. Want to see the different anticoagulants used majorly in hematology lab, whereby we shall have the name of the anticoagulant in one column. Then we shall have the color top. The color top. Then we shall have the mechanism of action of the anticoagulant. Then we shall also see the tests performed on blood which is stored in that anticoagulant, which is preserved by a particular anticoagulant. So this is the table I want us to form to summarize all the anticoagulants, whereby under each anticoagulant we are going to look at the name. Here we are looking at the name of the anticoagulant. After seeing the name, we are going to see the color of the top we use to cover. Then its mechanism of action, that is how does it prevent clotting, and the tests performed under each anticoagulant. And the major one we want to begin with is EDTA. And this EDTA in full is known as ethylene diamine tetra. So we can, it is internationally abbreviated as EDTA, what is called ethylene diamine. So this ethylene diamine tetra something. So this anticoagulant, for it is the one which is widely used in hematology. That is EDTA blood. So whenever you collect EDTA in a blood in EDTA tube, its color top is known as the purple top. You can always call it purple, or sometimes we call it lavender. That is the color code for it. You can call it the color top or the color code, which is lavender, or you can call it purple. And its mechanism of action of EDTA is that it acts by binding or chelating. Chelating means binding. Chelating calcium ions. But in an exam, avoid writing short forms, so we can always write it as calcium. Calcium ions. And we know that during blood clotting, calcium is very key to enhance blood, or to prevent, to enhance clotting of blood. So whenever we bind and we remove it from circulation of blood, now the blood cannot clot, because it inhibits the intrinsic and the extrinsic pathway of hemostasis. So after binding or chelating to calcium ions and they are removed from circulation, the blood remains the same. And these salts is made up of the EDTA salts. It contains of sometimes dipotassium. It has dipotassium, can have disodium, stroke, or tripotassium. But the most currently commonly used is tripotassium, which is the salt, and these salts are the ones that are preserving the morphology of cells. That when I'm to make a thin film, the cells remain the same shape even after 2 hours of collection. That is why we need these salts within the EDTA. And then these salts at the same time, they act by chelating or by binding together with these calcium ions. Then the tests performed, we see we can do them for performing blood slide tests. Blood slide, if we want to make blood slides for examination of hemoparasites, we can use EDTA blood. Or if we want to make a thin film comment, when we want to prepare thin film for film comment, we can use it. We can use it for cell count. Whenever we want to perform cell count, that is red cell count, platelet count, and white BC count, still we use this. That is, actually, this is what entails the full blood count, or what you call complete blood count. Whenever you want to do a CBC, you can always use EDTA. Then lastly is when you want to perform. So even here we shall see hematological indices. Hematological indices. We shall see PCV determination being done on these indices. We include MCV, MCH, MCHC. All of these are performed MCHC. All of these are performed on this blood. And lastly, we can do HB determination. But the whole of these ones, they entail what we call a complete or a full blood count. So if you want to do holistically a full blood count, now you are required to always preserve your blood in EDTA, which is responsible for doing this law. After finishing this EDTA, in the later video we shall see how can we preserve, or how can we prepare an EDTA tube. In the other video we shall look at that. And after seeing EDTA, the second one we can talk about is trisodium citrate, trisodium citrate. And this trisodium citrate is always come in concentration of 3.2 grams per liter. That is how it comes in. And this 3.2 gram per liter, its color code is light blue. Many students normally like writing blue, but it is not blue. When you say blue, you are wrong. We call it light blue. It is light blue. And the mechanism of action of this trisodium citrate is that it also acts by binding. It binds to calcium ions. And after binding to calcium ions, which are cofactors in enhancing clotting, whenever we bind and we remove it, when it is removed from the circulation, the blood cannot clot. So it acts by binding to calcium ions. After binding, it prevents clotting of blood. And this trisodium citrate, its importance majorly we perform tests like erythrocytic sedimentation rate, and the ratio we use is 1:4. 1:4, whereby 1 is for the anticoagulant and 4 is for blood. That is when you want to perform erythrocytic sedimentation rate, which is the rate at which cells, they form rocks, they aggregate and sediment within 1 hour. So you can always preserve them using 1 ratio of the anticoagulant and 4 parts of blood. Then if we want to perform coagulation tests, these coagulation tests, there are many of them, whereby they include the PT, prothrombin time, the APTT, the TTT, the thrombin time, and many others, D-dimers. We normally collect in a ratio of 1:9. That is 1 part of the anticoagulant and 9 parts of blood. That is how we preserve them, using 1 part of the anticoagulant and 9 parts of the blood. So these are the major tests that we can perform on a trisodium citrate, or blood preserved in trisodium citrate. Then another vacutainer we can talk about is heparin. And this heparin, we normally say, it is also naturally occurring in our body. So we can always ask you on objective that which of the following hematological anticoagulants can be naturally found in our body? That is heparin. So heparin is naturally found also in our body. Naturally found in the body. And that's why you see our blood, or your blood, does not clot, because the mast cells and the basophils, they produce this heparin, which prevents your blood from clotting within your body. But then we have extracted it and we are synthetically forming it to use it also to preserve our blood in the laboratory, whereby the color top is green, the code is green, and its mechanism of action is that this heparin, we normally say that if we have factor 10, activated factor 10, it can come and activate prothrombin to thrombin in presence of factor 5, in presence of calcium ions and platelet phospholipids. We are able to convert prothrombin to thrombin. And this thrombin is like a molecular scissor. It is the one that has ability to convert fibrinogen, which is factor 1, fibrinogen to fibrin, and this fibrin is a mesh. The mesh which causes clotting of blood. So what heparin does is that it is an antithrombin 3. Heparin is antithrombin. It is an antithrombin 3, meaning Since it is an antithrombin, it comes and sits on this reaction and prevents it from being converted from prothrombin to thrombin because it is an antithrombin. So being an antithrombin, it prevents formation of thrombin. And without thrombin, whenever I don't have thrombin in my blood, there is no cofactor that can convert fibrinogen to fibrin, which is the clot or a mesh of the clot at the injured site. So, due to the prevention of formation of thrombin, this one leads to failure of conversion. So there is no conversion or conversion of fibrinogen. In turn, it inhibits conversion of fibrinogen to fibrin, and without fibrin, the blood cannot form a clot. So this is how we can preserve the blood we have collected in the laboratory using heparin, whereby heparin is an antithrombin 3, which prevents conversion of prothrombin to thrombin, which is responsible for converting fibrinogen to fibrin, which is a clot. This one is a clot. So without thrombin, no fibrin, and no fibrin, no clotting. That is its mechanism of action. But after knowing its mechanism of action, the test performed under this is majorly osmotic fragility test. We can do osmotic fragility test, and we can also do RBC membrane enzymes, RBC enzyme estimation. But it is not commonly used in hematology lab. Why? This heparin, when used, and you want to make a film, when you stain a thin film with Roman whiskey stains, they tend to stain blue-black. So it is not the best anticoagulant used in hematology lab. We only use it when we want to do osmotic fragility test and red cell enzyme estimations. But its disadvantage is that when used, it causes staining of the cells to blue-black, which is not good for our film comment. Not good for the comment. Then the last one we can talk about, because we normally have major 4 anticoagulants used in the lab, and another one we can use is oxalates, but these oxalates are not a standalone. You cannot find oxalates alone. Oxalates like ammonium oxalate, we have potassium oxalate salts. They are always in combination with fluoride. So here we shall find fluoride oxalate bottles. So oxalates, they are not a standalone. They are always combined with sodium fluoride or lithium fluoride. So we can use either sodium fluoride or lithium fluoride. Then we combine them with oxalate salts, whereby these oxalate salts combined with the fluoride, the color code is always gray. We use gray color code, and these oxalates, for the oxalates in this bottle, they are responsible for binding loosely to calcium ions. They bind to calcium, they bind and remove calcium ions, stopping clotting, whereas the sodium fluoride, the fluoride is an inhibitor of an enzyme in glycolysis. So we say the fluoride is antiglycolytic. Fluoride is antiglycolytic. Antiglycolytic means it inhibits glycolysis. It is antiglycolytic, whereby it acts by inhibiting enolase. It inhibits the action of enolase enzyme. It inhibits the action of enolase. This enolase is a key enzyme in glycolysis, whereby it is the one that adds an eno group to 2-phosphoglycerate to form phosphoenolpyruvate, which later is converted to pyruvate, which is in a way of utilizing glucose. So without enolase, we cannot form pyruvate. We cannot form phosphoenolpyruvate, and without phosphoenolpyruvate, we cannot form pyruvate, which is always an end product of glycolysis. So whenever you collect blood, and I'm interested to determine glucose levels, I can always use fluoride to preserve blood in its normal concentration as in the body, because it inhibits glycolysis, which utilizes glucose in red cells. Then the hematological part of it is that we are utilizing oxalates, which is combined in the fluoride bottle to bind to calcium ions to stop clotting of blood, because we don't want the blood to clot. So they are used in combination, and we are seeing here, and we have been seeing that the tests performed is glucose estimation. Whenever we want to estimate glucose, we are using sodium fluoridated or fluoride oxalate bottles to preserve our glucose. So these are the major ones which we use in the laboratory. But the last one we can talk, I can even put it at this place, is the plain, which many students like most, which is number 5. It is the plain tube. But the word it says it is plain, it means there is no anticoagulant. So this is not for preserving. It is for just collecting blood for, and we do many tests like chemistry tests. But maybe why we can bring it here, but I cannot put as an anticoagulant tube because it is plain. So it is not an anticoagulant. It does not contain any anticoagulant. But we normally bring it in hematology, because sometimes in immunohematology, that is blood transfusion, we can always collect blood in a plain tube, and its color code is red. It's always red. It has a red top, and it doesn't contain, it does not have any anticoagulant. So whenever I ask you a question that discuss different anticoagulants used, and you find a student is writing for you plain tube, then you wonder. Because plain tube is the way the name suggests is plain, so it does not contain any anticoagulant, but it contains only some, these days, they put in clotting activators. So they normally add just clot activators. Why? Because we are not using anticoagulant, we need this blood to clot. So even add substances to enhance clotting. And this plain top, major in hematology, that is in immunohematology, that is blood transfusion, we are using it for cross-match. We are using it for cross-match, and cross-match, that is blood compatibility tests. That is what we can say. So majorly it is applied in immunohematology, which we call blood transfusion. And please don't forget, it is not among the anticoagulants. For it is not an anticoagulant, and we have said this, majorly we can do tests on cross-match, and other things is the chemistry tests. We can do in a plain top. Chemistry tests, and there are many majority of them which can be done. So these are the anticoagulants, the major 4 anticoagulants that we are using or utilizing in the laboratory to preserve our blood. And after we have known different anticoagulants, the color codes, the mechanism of action, and the tests performed under each. Now what we need to know is the qualities of a good anticoagulant. How can you say that this is a good anticoagulant? Number 1, a good anticoagulant should be able to preserve the morphology of cells. The morphology of cells should remain the same. That is what we call a good anticoagulant, meaning it should not cause shrinkage of cells. And another good quality is that they should not cause hemolysis. They should not cause hemolysis, meaning the cells should remain in their normal shape without hemolyzing. So there are some anticoagulants that can enhance hemolysis, but a good anticoagulant should not cause hemolysis, should preserve the morphology of cells, should be able to prevent shrinkage of cells, especially red cells. And another property, you stop it.